looking at, we're going to continue with chapter 25 and going on part 3 of part 4. So now we're going to look at the liver, the gallbladder, the pancreas, which are our accessory organs, and then the small intestines. Okay, so we'll look at the liver. Oh, the small intestines. They're the ones that's going to receive the chyme. So now we've left the stomach, well, we've left the esophagus, they left the stomach. It's considered to be chyme now, and it's on its way into the small intestines. Now they're the ones going to receive the chyme from the stomach. Now, when it's in the stomach, it's going to be when it's in the small intestines, it's going to be receiving secretions from the liver as well as from the pancreas. Now these secretions are going to enter the digestive tract near the junction of the stomach and the small intestines, so that area right in here. Now these secretions are important to the digestive process of the small intestines. So let's look at the liver, and then we'll get back into the small intestines. So this liver, you guys should be very familiar with it. It's reddish in color. It's located near to the diaphragm, and it is the body's largest gland, weighing approximately 1.4 kilograms or 3 pounds. Now it has many functions, of which only one, the secretion of bile, contributes to digestion. Now it has 4 main lobes. When we look at these 4 lobes, we have a right, a left, a quadrate, and as well as a caudate. Now anteriorly, the right and left lobes are visible and are separated from each other by the phallicoform ligament, which we see right here. A phallicoform ligament. We have our left lobe and our right lobe that are divided or separated by this folliculiform ligament. Now the round ligament, also called the ligament teres, is also visible anteriorly and is a remnant of the umbilical vein. Now from the anterior view, the quadrate lobe, here we go, the quadrate lobe is seen next to the gallbladder, and the tail-like quadrate lobe is posterior to that. Now we have an irregular opening between these 2 lobes. The porta hepatis, which is a point of entry for the hepatic portal vein, and the proper hepatic artery, which we'll see in a different diagram. Here we go. Here's our hepatic artery, and of course our portal vein. Now the gallbladder, which you see right here in green, it adheres to depression on the inner surface of the liver, and is between the right and the quadrate lobes. Posteriorly, the liver, it exhibits a deep groove called a sulcus that accommodates the inferior vena cava. Now the superior surface has a bare area where it attaches to the diaphragm. And the serosa covers the rest of the liver. So here is our right lobe, here is our left lobe, and in the middle that separates this ligament that separates the 2 is called that round ligament. No, that separates the 2 is our folliculiform ligament, and at the tail end of it is our round ligament. Looks like an umbilical cord. And you see a little tip, this green area right here, this will be our gallbladder. This is our anterior view, and at the top you see this inferior vena cava. In the posterior view, we see it a lot better, where we see the superior vena cava, and that little deep groove, or the sulcus area, sulcus which is a deep groove within the within the liver, is what holds the inferior vena cava, which is that blood vessel that will take the blood towards the heart. Right, and also we see in the porta hepatis, we see the hepatic portal vein, the hepatic artery proper, as well as the bile duct, and of course our bile. And this area is our a quadrate lobe. We have our right lobe right here, and here's our bigger area, and our caudate lobe. So let's go ahead and look at the microscopic view or microscopic anatomy of the liver. Now, the interior of the liver is filled with these tiny cylinders called hepatic lobules, and they're about 2 millimeters long by 1 millimeter in diameter. Now, lobus consists of a central vein surrounded by radiating sheets of cuboidal cells called hepatocytes. Let me get this right here. There are hepatocytes. All right. Okay. Now, each plate of hepatocyte is an epithelium, one of 2 cells thick. Now, the spaces between these plates are blood-filled vesicles called hepatic cyanoids. Okay, here we go. Here's hepatic cyanoid right here, and they're lined with fenestrated endothelium. Sorry. Now the hepatocytes have a brush border of microvilli that project into the cyanoids. Now, blood that filters through the cyanoids, they come directly from the stomach and the intestines. The hepatocytes, they absorb nutrients from this and also remove and degrade hormones, toxins, and bile pigments and drugs. The hepatocytes also secrete albumin, lipoproteins, Claudin factors, angiotensinogen, and other factors in the blood. Between meals, they also break down stored glycogen. Now, the cyanoids also contain hepatic macrophages, also called Kupffer cells. Now, and they are going to remove any type of bacteria or debris that's left inside of there. Alright, so let's jump ahead to our bile canaliculi. Now, the liver is going to secrete bile into these narrow channels called as bile canaliculi. And they're between the back-to-back layers of the hepatocytes within each plate. So here we have hepatocytes, and here we have the bile canaliculi. I keep saying canaliculi. Canaliculi, which are these small little channels that you see here, where the bile is going to be secreted out from the liver. Okay, so bile is going to pass through those particular ducts of triads, and then ultimately into the right and left hepatic ducts. Now, the hepatic ducts, they converge on the inferior side of the liver to form the common hepatic duct. Now, a short distance further on is going to join with the cystic duct coming from the gallbladder. Now, the hepatic lobes, they're going to be, well, let's stick with this one. Near the duodenum, the bile duct is going to join ducts of the pancreas. Forms expanded chambers are called the hepatopancreatic ampulla, and they seem to terminate in a fold of tissue. Now, the major duodenal papillae, they consist of muscular hepatopancreatic sphincter, also called the sphincter of Ode or of Ode. And they're going to regulate the passage of bile and the pancreatic juice into the duodenum. Now between meals, this sphincter is going to close and prevent the release of bile into the intestines. So here I have a picture of the common hepatic duct. We have our cyst duct coming directly from the gallbladder, and they're going to join up and form that bile duct that will secrete into the small intestines. Well, yes, small intestines, which would be that small area of the duodenum. Okay, so let's look at the gallbladder now. Gallbladder, another one of the accessory organs, is a pear-shaped sac on the underside of the liver, and it's going to it stores and concentrates the bile. Now it is about 10 centimeters long and is lined with a highly folded mucosa with a simple columnar epithelium. Now its head is considered to be the fondest, and it usually projects slightly beyond the inferior margin of the liver. Okay, so you see that the picture has changed. I went ahead and just added a picture so you guys see exactly what I'm referring to when you look at this pear-shaped organ in the body, the gallbladder. And we look at the different types, the different areas of the gallbladder itself. So we're talking about the head. This is the head right here, or also called the fundus region. And this is the one that the book talks about, and it says how it projects slightly beyond the inferior margin of the liver. And this area up here is the neck, or the cervix, is what leads to the cystic the duct. So now you can see exactly what it looks like, especially the length, how it's approximately 10 centimeters in length. Okay, now you get a visual of what everything is looking like. Okay. All right. So what does it secrete? It secretes bile, and bile is this yellow-greenish fluid that contains these minerals, the cholesterol, the neutral fats, the bile pigments, the phospholipids, as well as the bile acids. Now the primary pigment is the bilirubin, and is derived from the composition of hemoglobin. Now bacteria of the large intestines, they're going to metabolize the bilirubin to urobilirubin, which gives feces that dark color. So here we have the bilin, and they're going to be metabolized to urobilirubin. Bilinogin, sorry. And that's what gives the feces that dark color. Now, in the absence of this bile secretion, the feces are grayish white and marked with streaks of undigested fat, which would be that alcoholic feces is what they'll call it. Now the bile salts, also called bile acids, they're steroids synthesized from cholesterol that, together with the lecithin, they aid in fat digestion as well as absorption. So these bile salts, along with the lecithin from the cholesterol, synthesized from cholesterol, along with the lecithin, they're going to aid in fat digestion and with fat absorption. So altogether, these components of bile are wastes designed for excretion. And if these wastes become excessively concentrated, they may form gallstones. And everybody should be familiar with what gallstones are. Okay. Now bile gets into the gallbladder by first filling the bile duct, then overflowing into the gallbladder. Now, between meals, the gallbladder is going to absorb it absorbs water and electrolytes from the bile, and it concentrates it by a factor of 5 to 10 times. Now, the liver, it secretes about 500 to 1,000 milliliters of bile per day. Now, about 80% of the bile acids are reabsorbed in the ileum, which is that last portion of the small intestines, and are returned to the liver, where the hepatocytes, they're going to absorb them, they're going to resecrete them, and that process is called enterohepatic circulation. Now, this process, it reuses the bile acids 2 or more times during the digestion of a meal. Now, the 20% that is not absorbed is secreted in the feces. This is the body's way of eliminating excessive or an excess of cholesterol. The new bile acids are synthesized from cholesterol to replace the quantity lost in the feces. Alright, so let's look at a couple of terms here. We have gallstones and lithotripsy. These are some terms that you will see again, such as the formation of gallstones. We have the hard masses in either the gallbladder or the bile ducts. Those are the gallstones, and composed of cholesterol, calcium bicarbonate, as well as bilirubin. And this term here, which deals with the formation of gallstones, is the one that it's most common in obese women over 40 with an excess of cholesterol. We have also a painful obstruction of ducts, and that's in a result of jaundice, poor fat diet or digestion, and impaired absorption of fat-soluble vitamins. And we have lithotripsy, which is the use of ultrasound vibration to pulverize stones without surgery. So if someone has gallstones, they'll use this particular procedure to sort of break up the stone, which is called pulverize, break up the stone so that it can be released. Alright, so we're here onto the kidneys, which sits very close to the stomach, as well as very close to the liver. It is a spongy gland that's posterior to the greater curvature of the stomach. So here's our greater curvature of the stomach. It sits behind that part, which is posterior. It's approximately 12 to 15 centimeters long and 2.5 centimeters thick. It has a few areas. It has a body, and that body is in that midsection. It also has a blunt, tapered tail on the left-hand side. It is both endocrine and an exocrine gland. The endocrine part involves the pancreatic islets, which secrete the insulin and the glycogen. Now, about 99% of the pancreas is exocrine, which secretes 1,200 to 1,500 milliliters of pancreatic juice per day. Now, the cells of the secretory acini it exhibits a high density of rough ER and the zymogen granules, which are vesicles filled with secretion. Now, the acini, they open into the system of the large ducts that eventually convey on the main pancreatic duct. which you really cannot see those small ones there, but that's the, here's our pancreatic duct here. Okay. Now the accessory pancreatic ducts, or duct, however, they branch from the main pancreatic duct and open independently into the, here we go, into the duodenum at the minor duodenum papillae, which is posterior to that major papillae. Now the accessory duct allows the pancreatic juice to be released even when the bile is not released. Now the pancreatic juice is an alkaline mixture of water, enzymes, zymogens, sodium bicarbonate, and other electrolytes. Now the acini, they secrete the enzymes and the zymogens, while the ducts secrete the sodium bicarbonate. Now the bicarbonate, it serves to buffer this hydrochloric acid arriving from the stomach. Now the pancreatic zymogens are trypsin, chymotrypsinogen, and the procarboxyl peptidase. Those are the three pancreatic zymogens. Now, when secreted into the intestinal lumen, the trypsin is converted to trypsin by this particular enzyme called the enterokinase, which is secreted by the intestinal mucosa. Now, the trypsin is autocatalytic, and it converts the trypsin into more trypsin. And the trypsin also converts the two other zymogens into chymotrypsin and the carboxy peptidase. Alright, so to kind of put this in perspective as to what's going on with this particular zymogens, I'm going to draw a little diagram for you guys to help you out with identifying the function of the purpose of this this trypsin. Now we already talked about the three pancreatic zymogens. Okay, let me see if I can get my drawing going. Alright, so we talked about the three of them. If you guys look back in your books, is the trypsinogen, the chymotrypsinogen, and the procarboxy peptidin. So I'm going to initial it as, we have our T, we have our C, and we have our P, and those are for three pancreatic zymogens. So what I was talking about, how when it's secreted into the intestinal lumen, the T, that's my my trypsinogen, they're going to be converted, or it's going to be converted into trypsin. And the enzyme that assists with that is, well, let me see if I can get the color for that. Is enterokinase, E-N-T-R-O kinase. Remember, whenever you hear the word ase, or the ending, the suffix ase, we're talking or referring to an enzyme. And that enzyme basically is secreted by the intestinal, I'll do an abbreviation, intestinal mucosa. ISA. Okay. Now this trypsin that is formed, it has a number of functions, or it has plays a huge role. And what it does, when it says it's an autocatalytic, it means it's going to help in breaking it down again. It's going to help break down, and it's going to help with converting this the trypsinogen again into more trypsin. So it's basically going to help in converting this T into more of itself. It wants to make more trypsin, alright, so what it's going to do, help with breaking down this the trypsinogen back into trypsin. So it's going to work in tripling, quadrupling itself. What else does it do? The second pancreatic zymogen, which is our C, our oh my word, these pronunciations. The chymotrypsinogen, our C, what it does with that, it helps with converting okay, and what else it does here, it converts the remaining two zymogens into other factors that play a role. So what it does, the C, this trypsin helps convert the C into our T. Of course, it will make more trypsin. As well, it helps in converting it into chymotrypsin, which is CHYMO trypsin. Okay. This trypsin also helps convert our P, which is our pro-carboxypeptidase, helps convert it into carboxypeptidase. So you see that this trypsin plays a huge role in ensuring that we have enough trypsin for digestion. Alright, so let's get back to our original screen. Okay, so that's dealing with these pancreatic zymogens. So we have other pancreatic enzymes. We have the pancreatic allomease, which is there to digest our starches, our breads, our carbohydrates. Then we have the pancreatic lipase, which digests our fats. We have ribonuclease and deoxyribonuclease, which digests the RNA and our DNA respectively. Alright, so here's just a small, another diagram here to show you what's happening when the trypsinogen is secreted or released into the small intestines, how this trypsin helps in breaking it down, with the help, how the trypsinogen is broken down into trypsin with the help of the enterokinase, and how the trypsin comes back around to make more trypsin by converting the trypsinogen into more trypsin. The same diagram I did earlier. and of course helping this other enzyme again broken down into another product. Alright, so now we have some stimuli. Now, secretion is regulated by responses that cause release of pancreatic juices and bile upon three types of stimuli. We have the acetylcholine, we have our CCH, and we have our secretin. Now, the acetylcholine coming from the vagus and enteric nerves, they're going to stimulate the pancreatic acne, and they're going to secrete these enzymes even before food has been swallowed. Now, these enzymes are going to remain stored in that pancreatic acne duct and the ducts, however, for release when chyme enters the duodenum. So it's going to be stored until they feel that until that chyme is there in the duodenum. Now, the CCK, they're secreted, or it is secreted by the mucosa of the duodenum and the proximate duodenum in response to fats in the small intestines. Now it stimulates the pancreatic acne to secrete its enzymes, and it has a strong stimulatory effect on the gallbladder, from which it gets its name. It induces contractions of the gallbladder and the relaxations of the hepatopancreatic sphincter. Now, the third one, our secretin, is produced in the small intestine in response to the acidity of chyme from the stomach. Now, it stimulates the ducts of the liver and pancreas to secrete sodium bicarbonate to offset the hydrochloric acid, and then to protect the intestinal mucosa from any type of stomach acid. So the secretin, they're there to stimulate the ducts of the liver and the pancreas, so they can secrete this HCl, the hydrochloric acid, so that it can offset the hydrochloric acid from being released, so that it can coat the stomach and make sure that the inside of the intestinal mucosa is protected from the stomach acid. Alright, so let's move on to the small intestines. Small intestines, we know it has a number of sections, it has a certain size, but we want to get into, of course, this with anatomy, we're going to deal with the structure of it as well. Alright, so nearly all chemical digestion and nutrient absorption occur in the small intestines, so called because of its small diameter. It is the longest part of the digestive tract. And in terms of the gross anatomy, when we look at it, it is a coiled mass filling most of the abdominal cavity, inferior. Here's the stomach. So it is inferior to the stomach and the liver, and is divided into three regions: the duodenum. Let me get this mouse working. Here we go. The duodenum, the jejunum, which is that purplish color, and then we have the ileum, which is that pinkish area. Now, when we talk about the duodenum constitutes the first 25 centimeters or 10 inches of the intestinal tract, and it begins at the pyloric valve. Now it arches upward. Let's go back to the diagram. Now what it does, it's going to arch upward, and it has wrinkles called the major and the minor duodenal papillae, which you cannot see on the outside, well, in this particular diagram. And this is where the pancreatic duct and the accessory pancreatic ducts will enter in this particular area. All that stuff we were talking about with the pancreas and everything being secreted, this is the area that you'll have all the secretion taking place. Not in the duodenum, not in the ileum, we're talking about the pancreas. This is the region. Now, the name duodenum refers to its length, about that of 12 fingers. Now, it's slightly distal of the pyloric valve. Now, of course, it has its wrinkles. Now, most of the duodenum, along with the pancreas, is the retroperitoneal. Now, here the stomach acid is going to be neutralized. The fats are going to be emulsified by the bile acids, and of course, our pepsin is going to be inactivated by the increase in the pH levels. And of course, our pancreatic enzymes will take over after chemical digestion. Alright, so now our duodenum is the first 40%. The first 40% of the small intestine beyond the duodenum. Now, its name refers to the fact that early anatomists, they typically found it to be empty. Now, it begins in the upper left quadrant of the abdomen, but lies under or lays mostly within the umbilical region. It has large, tall, closely spaced closely spaced circular folds with relatively thick and muscular walls and a rich blood supply, giving it its relatively red color. Now, most digestion and nutrient absorption will occur here. Now, when we look at the ileum now, it forms the last 60% of the postal duodenal small intestine. So we're looking at it being about 1.6 to 2.7 meters. Now, it occupies mainly the hypogastric region and part of the pelvic cavity. Compared with the duodenum, its wall is thinner, less muscular, and less vascular, and it has a paler pink color. On the opposite side, from its mesenteric appearance, which is on the back side, which you can't see, the ileum has prominent lymphatic nodes in clusters called Peyer's patches. Now, the end of the small intestine is the ileocecal junction, and that's where the ileum is going to join with the cecum of the large intestine. Now, the muscularis of the ileum is thicker or thickened here to form a sphincter called the ileocecal sphincter valve, which is going to protrude into the cecum and then regulate the passage of food residue. Now, both the duodenum and the ileum are intraperitoneal and thus covered with the serosa that is continuous with the folded mesentery that suspends the small intestine from the posterior abdominal wall. Now, when we continue to look at the layers of the small intestines, we notice that it will have a lumen, and this lumen is lined with the simple columnar epithelium. Now, the muscularis externa is obviously the thicker inner circular layer and the inner outer longitudinal layer. Now, the large internal surface area is produced by the small intestine's relatively great length and by three kinds of internal folds or projections. We have the circular folds, which are the largest folds and are transverse to the spiral ridges. We have the villi, which increase surface area by a factor of 10. and they actually look like little fingers or tongue-like shapes. And then we have the microphyllae, and they increase surface area by about 20%. By 20. I'm sorry. I'm gonna go to this particular diagram here so you can get a good look of the villi. Sorry, here's the villi that look like fingers. Of course, they are the largest in the duodenum, and then they begin to get smaller as the regions within those distal regions. So they'll look a little bit smaller in the jejunum as well as in the ileum regions. Now these villus. Now a villus, which is the plural form of villi, you have one villus, 10 villi. They're covered with two kinds of epithelial cells. You have the columnar obstructive cells and the mucus secreted goblet cells. And these cells are joined by tight junctions, and they prevent digestive enzymes from seeping between them. Now the core of the villus is filled with areolar tissue of the lamina propria. Now we do have blood vessels, which we see here in the diagram, blood vessels that absorb most of the nutrients. But the lacto, get here the green one here, the lacto, this is the one that's actually going to absorb most of the, here we go, most of the lipids. Now the core of the villus also contains few smooth muscle cells that contract periodically to mix the chyme and then to move lymph down the lacto. Let's go back here. Alright, so each absorptive cell of the villus, they're going to have these fuzzy looking branches or brushes called the microphyllae, and they're about 1 micrometer high. So that's not very high at all. You only can see them underneath the microscope. Now the brush border, they're going to increase the surface area, and they're going to and it contains brush border enzymes in the plasma. Now these enzymes are not released into the lumen, but digest the chyme contents as it contacts the brush border, and that's a process called contact digestion. Let's move on because I know it's a lot of information. Let's look at the 3 functions, contraction functions of the small intestines. They are to mix chyme with the intestinal juice, bile, and the pancreatic juice, and thus is to make sure that is to neutralize the acid as well as digest nutrients more effectively. Second one, to churn the chyme and bring it in contact with the mucosa for contact digestion and nutrient absorption. And the last function of the small intestine contraction is to move residue toward the large intestines. Alright, for segmentation, I want you to look at that diagram. Here's the information here that we're going to talk about. But I'm going to turn to your book, or if you have the book open, or you can just look at the diagram there. Figure 25.26a shows you a diagram of segmentation, and that deals with the movement in which stationary ring-like contractions appear at several places along the intestine and then relax as new contractions form elsewhere. Alright, so the purpose of this segmentation is to basically to mix, the churn, and not to move materials along as peristalsis, because peristalsis is just the wave-like movements, but this is totally different for peristalsis. And they get their rhythm by these pacemaker cells of the muscularis and externa, which is one of the layers, at about 12 times per minute in the duodenum, and then 8 to 9 times per minute in the ileum. Now, the slower the rate in the distal regions assists with progression toward the colon. The intensity of the contractions is modified by the nervous and the hormonal influences, but not by the frequency. So peristalsis. Peristalsis, it moves contents of small intestine towards the colon. Now, the peristaltic wave begins in the duodenum, and it travels 10 to 70 centimeters, and then it dies out, only to be followed by another wave that begins a little further down the track. Now, these successive overlapping waves are called migrating motor complex. Now, over a period of about 2 hours, they move the chyme toward the colon. A second complex then expels the residue and bacteria from the small intestine, thereby helping to limit bacterial colonization. Now, the refilling of the stomach at the next meal suppresses the peristalsis and reactivates segmentation. Now, the ileocecal valve is usually closed, but food in the stomach is going to trigger both the release of the gastrin and the gastroileal reflux, both of which are going to enhance that segmentation in the ileum, and then it's going to start to relax that valve. Now, as the cecum starts to fill, the pressure, it pinches the valve shut, and it prevents the reflux of the cecal contents into the ileum. Alright, so yes, it is a lot of information that we've covered so far in parts 1, 2, and 3, going into grave detail with the various parts of the digestive system. But it has to be covered, and you guys have to be very familiar with this information. So in the next part, well, in the final part of chapter 25, we're going to look at the remaining parts of the digestive tract, the large colon and the rectum area. Okay. And then we're going to look at chapter 26. I think I have a few videos for you for that. Okay. So if you have any questions, again, post it on Blackboard in the discussion area, and I'll be able to answer them for you. Go ahead and start looking at part 4.